My name is Adil Akif. I'm a PhD student in the Hydro Lab over at Yale University, and today I'll be talking about our efforts to predict neuropil density from MRI images. And so when I talk about neuropil density, we're referring to neuronal and glial cell bodies and their filaments which end in synapses. So one way to characterize neuropil density is to look at cell density covering both neurons and glial cells, as well as synaptic density. Now, the neuropil is electrically active, and this is starting to come into the interplay between structure and function in the brain. And one way we can quantify this electrical activity is to look at average neuronal firing rates. And given the neuropil density and the electrical firing rates, we're able to better understand metabolic heterogeneity throughout the brain, because regions of the brain that have more neurons, more synapses, or higher firing rates will require more energy. And we can model this using something called the energy budget. And now this next slide goes into this a bit more graphically. So when we're doing an energy budget model of the brain, we know the energetic costs for different processes that occur on a cellular level. For example, housekeeping would include things like protein synthesis or synaptic modification, while the action potential component would cover neural signaling. So we take these different energetic costs that we are able to deduce from values in the literature and through various calculations, and we have to scale them by the appropriate parameters. So some of them scaled by cell density, some of them synaptic density, as well as some of them by electrical activity. And so when we're running this model, we use a single firing rate for electrical activity. We add up the different energetic components, and we're able to get a prediction for energy consumption and energy map of the brain, which we can compare to in vivo images from a modality like calibrated fMRI. Or if you like, we can run this model in reverse, so to speak. We can use the calibrated fMRI data as an input and turn the electrical activity as a free parameter, giving us a means for calculating electrical activity throughout the brain. And this is just another visualization of the same principles, as well as some summary statistics over here for gray matter and white matter throughout the brain. But what I really want to focus on here is neuropil density. How do we acquire the cell density and the synaptic density needed for our energy budget? So until now, we've just been using prototypical values. We have we've gotten cell density from the BigBrain dataset, as well as synaptic density from PET imaging. And so the BigBrain dataset's a 3D histology with marker staining of the brain, and the intensity of this stain can give us a proxy for cell density throughout the brain. As for synaptic density, we're able to use a PET tracer, UCBJ, which targets synaptic vesicle glycoprotein 2A, which is expressed throughout synapses in the brain. So this gives us a means for imaging synaptic density. And now going back to our energy budget model, we see the cell density and synaptic density parameters over there. We do have prototypical values for this, but what we were trying to do in this study is try to find out, is there a way to acquire these values from MRI data? And so I'll go through this quickly, but these are some of the data sets that we've been using. So MP-RAGE, or T1 imaging, is able to give us a superior contrast between gray matter and white matter. And the understanding here is that depending on the tissue we're in, this is going to influence the neuropil architecture. And the neuropil architecture, in turn, will determine the diffusion properties of the tissue, which can be captured through metrics like apparent diffusion coefficient or the directionality of diffusion, fractional anisotropy. And all of these things are metrics that we can measure using MRI. So our hypothesis was that we could use this as a starting point to calculate cell density and synaptic density throughout the brain. And so these different data sets we have acquired from the ADNI database, which is a multi-center imaging study in the United States. We focused on the control subjects, and 9 subjects met our criteria. And so these are the 9 subjects over here. We have the MP-RAGE, ADC, and fractional anisotropy data sets from MRI imaging. And you can put this through different levels of Gaussian smoothing as well. And that's important to give our neural network some neighborhood information. So this over here is the neural network we use for training. We provide, it works on a voxel by voxel basis. So it's not a convolutional neural network. We'll provide the ampere age, ADC, fractional length, anisotropy at a voxel, as well as about 12 other data points as we put these data sets through different levels of Gaussian smoothing. And this goes into the network. It's a fully connected network with 3 layers, 40, 20, 10 neurons, and the output is cell density or synaptic density depending on what we're training it for. Now, unfortunately, we were limited in the data sets at this point because we do not have the cell density or the synaptic density for each of these 9 different subjects. We just have the prototypical values from the BigBrain and also a group average synaptic density. And so, in a way, this was a feasibility study to see whether it made sense to try to acquire higher quality data sets, better matched data sets to do training as a follow-up. And so the first attempt over here was a group average training. So on the top left, you can see the group average the synaptic density data set. That's our gold standard. For this network, we did group average to group average training. So we averaged the 9 ADNI subjects and used the group average synaptic density as output. And you can see the individual predictions on the second row. So it's not predicting the kinds of patterns we want to see. We do not see the distinction between gray matter and white matter that we see in our original data set. So this is the second training approach we used. We still do a group average to group average training, but we provide less smoothing to the neural network. So now it's being forced to train on the variation you see within the group averages. And by doing so, the neural network is able to better generalize when it has to predict on the different individuals. So on the second row, you see much more reasonable predictions, but the data is still one of the things we do compromise over here by reducing the smoothing given to the neural network is that the images, they look noisier. They sort of look a bit more grainier. You have less spatial autocorrelation compared to the gold standard. And so instead of reducing the smoothing provided to the network, another thing we do is individual training. And so this was a bit strange because of the limitation in our data set, but what we tried over here is we took the 9 different subjects and assumed the same synaptic density gold standard as an output, just to see whether this would be a feasible approach going forward. And so you can see over here that we do end up seeing the smoothness in the data and more reasonable predictions in terms of synaptic density. And we do the same thing with cellular density. Group average to group average prediction with high smoothing does not generalize well to the individuals. When you provide it less smoothing and force it to train a bit more rigorously, it generalizes a bit better. But finally, when you do individual training but maintain that high smoothing, we get much more reasonable predictions. In this particular case, the neural network seemed to struggle a bit in the predictions, trying to predict the same BigBrain neuropil density regardless of subject. But we wanted to see whether it was learning any useful relationships. And so in the final row, you can see the normalized data set with the standard deviation boosted. And we do see the kinds of distributions and a similar level of smoothness to our gold standard. And so just a summary, group average training with highly smooth input does not give us the kinds of spatial distributions we see in the gold standard. When we do group average training without the smoothing, we do see the expected spatial distributions, but not the expected spatial autocorrelations. And when we do the individual training, we end up getting the expected kinds of images. And so we consider this the best performing option. And so going over some of the limitations, in the future, ideally, we would acquire cellular density data for each of our different subjects for which we have ampere age and diffusion data. And so this can potentially be done through MR methods like NADI and MADI. And in the future as well, any subject undergoing these MR scans can also be put through PET scans to acquire synaptic density. And with a higher quality data set like this, the presumably the prediction accuracy should not come at the cost of overfitting. So we can try to use more powerful neural networks, such as convolutional neural networks. The focus of the study was to look at the simplest network architecture that could give us reasonable results, and that seemed to be the shallow network with those predefined kernels. And finally, I'd like to acknowledge my research committee, the Hyder Lab, and the co-authors on the study.
Then I take the opportunity. Actually, when you started talking, and you were going into the details, cellular organization, glial apparatus, etc. I thought actually that you're going and start talking about timescale evolution also, accompanying neural activity. But am I correct that time, temporal, the slow evolution of the energy household is not any of your concern, right?
Well, that is something we are looking into. So, okay, yeah, so that is actually one of our main goals. We eventually do want to the slides aren't up, but we eventually do want to be able to add a dynamic aspect to this. So with calibrated fMRI, once you have the calibration down, you can get dynamic fluctuations in metabolism, and if we plug this into our model, we should be able to get dynamic firing rates as well. And so this is one of the things that we're working towards.
Okay, very important in fact and very promising. Thank you very much. Yeah?
All right.